Hey kids, this is Cookie Monster. Mrs. Cookie Monster is learning about cell division. Cell division, when one cookie breaks to two, make more cookies. Cell division make Cookie Monster happy. Right kids?
This is how we do it! This is how we do it!
Today I'm going to talk to you about what happens in the cellular processes of mitosis and meiosis. Mitosis is the division of a body cell, a somatic cell, to form two daughter cells, each with the same chromosome complement as the parent cell. Before mitosis can occur, the cells are in interphase, during which the chromosomes are decondensed, less tightly compacted, and found in the nucleus. After interphase, mitosis is then broken down into 5 subphases: prophase, prometaphase, metaphase, anaphase, and telophase. At the start of prophase, the chromosomes have already replicated to produce 12 chromatids. The nuclear membrane begins to dissociate into small vesicles. Chromatids condense into more compact structures, and the mitotic spindle begins to form as the nucleolus disappears. In prometaphase, the centrosomes move to opposite ends of the cell and demarcate 2 spindle poles, one within each of the future daughter cells. Once the nuclear membrane has dissociated into vesicles, the spindle fibers can now interact with the sister chromatids. The kinetochore microtubules are rapidly formed and can be seen growing out from the 2 poles. The kinetochore on a pair of sister chromatids is attached to the kinetochore microtubules from the opposite poles. The sister chromatids are seen to undergo jerky movements as they are tugged back and forth between the 2 poles. By the end of prometaphase, the mitotic spindle is completely formed. In metaphase, the sister chromatids align themselves along a plane called the metaphase plate. At this point, each pair of chromatids is attached to both poles by kinetochore microtubules. The pairs of sister chromatids have become organized into a single row along the metaphase plate. After the chromatids have lined up, they are then equally distributed into 2 daughter cells. In anaphase, the connection responsible for holding the pairs of chromatids together is broken. Each chromatid is now an individual chromosome and is linked to only one of the 2 poles. The chromosome moves toward the pole to which they are attached. This involves the shortening of the kinetochore microtubules. The 2 poles themselves move further apart due to the elongation of polar microtubules. These slide in opposite directions due to the actions of motor proteins. In telophase, the chromosomes reach their respective poles and decondense. The nuclear membrane now reforms to produce 2 separate nuclei. The nucleoli reappear, and in cytokinesis, the 2 nuclei are segregated into separate daughter cells. Now let's go over the process of meiosis. Meiosis involves 2 successive divisions rather than 1 as in mitosis. Before meiosis can occur, the chromosomes are replicated in the S phase to produce pairs of sister chromatids. In prophase 1, replicated chromosomes begin to condense. Each structure is a pair of chromosomes. Synapsis, the process in which the homologous chromosomes recognize each other and then align themselves along their entire length, occurs. Crossing over also occurs. Crossing over involves a physical exchange of chromosome pieces. In prometaphase 1, the spindle apparatus is complete and the chromatids are attached via kinetochore microtubules. In metaphase 1, the tetrads are all arranged at the metaphase plate with 1 chromosome facing each pole. Microtubules from one pole are attached to the kinetochore of 1 chromosome of each tetrad. Bivalents independently align themselves at the metaphase plate of the spindle. Maternal and paternal homologs of each bivalent may be oriented toward either pole. In anaphase 1, the homologous chromosomes separate. 1 chromosome moves toward each pole guided by the spindle apparatus. The sister chromatids remain attached at the centromere and move as a single unit toward the pole. Each chromosome still has 2 chromatids. In telophase 1, the movement of homologous chromosomes continues until there is a haploid set at each pole. Each chromosome consists of 2 sister chromatids. The nuclear envelope reforms and nucleoli reappear. Cytokinesis usually occurs at the same time by the same mechanisms as mitosis. In animal cells, a cleavage furrow forms, whereas in plant cells, a cell plate forms. Mitosis 2. In prophase 2, a spindle apparatus forms and attaches kinetochores of each sister chromatid. The spindle fibers from one pole attach to the kinetochore of one sister chromatid, whereas those of the other pole attach to the kinetochore of the other sister chromatid. In metaphase 2, the sister chromatids are arranged at the metaphase plate. Because of crossing over in meiosis I, the two sister chromatids of each chromosome are no longer genetically identical. The kinetochores of sister chromatids attach to microtubules extending from opposite poles. In anaphase 2, the centromeres of sister chromatids separate and two newly individual chromosomes travel toward opposite poles. In telophase 2, the chromosomes arrive at opposite poles. Nuclei form around the chromosomes, which begin expanding, and cytokinesis separates the cytoplasm. At the end of meiosis, 4 haploid daughter cells are made.
Thanks, kids. Now Cookie wants to know what cell division is.